All right, today we're diving into a really fascinating, kind of complex medical puzzle called amyloidosis. The best way to think about it is like a detective story, but one that's happening right inside the human body. The clues? They're super confusing. And the culprit? Totally microscopic. So, let's get our detective hats on. Our investigation starts right now. So, the wild thing about amyloidosis is that it almost never starts with a clear diagnosis. Instead, it begins with this collection of symptoms that seem totally random. You know, a patient might be bouncing around between different specialists for all these separate issues, and nobody's putting it all together. It's like a classic case of something hiding in plain sight.
Okay, so let me ask you this. It's kind of like a riddle. What on earth could carpal tunnel syndrome, heart failure, and weird bruising around the eyes, they call them raccoon eyes, what could they possibly have in common? I know, it sounds like there's no connection, but the answer is, they can all be clues pointing right to amyloidosis.
And that's just the start. The trail of clues here is it's all over the place. We're talking about nerve problems that cause numbness in your fingers and toes, a heart that gets all stiff and can't pump right, problems with your kidneys, even an enlarged tongue. Oh, and getting dizzy every time you stand up. I mean, on their own, any one of these could be 100 different things, right? But when you see them together, well, that's when a really sharp-eyed medical detective starts to see a pattern.
So what is it? What's the single culprit behind this whole baffling mess of symptoms? Well, the answer isn't a virus or some kind of bacteria. The answer is way smaller, down at the molecular level. It's actually something our own bodies make. It's a protein that's gone completely rogue.
And here it is. This is the official definition, kind of like the culprit's rap sheet. Amyloidosis is what happens when perfectly normal proteins in our body just misfold. They literally get bent out of their proper shape. And instead of doing their job and then being cleared away like they're supposed to, they get sticky. They start clumping together and forming these tough, stringy fibers that build up in our organs.
Let's walk through how this happens. It all starts with one tiny error. You've got a normal, happy protein floating around, but then it takes a wrong turn and folds into a weird, abnormal shape. This new shape makes it sticky, so it starts glomming on to other misfolded proteins. These clumps just keep growing and growing, forming these long, tough fibers called fibrils. And these fibrils are what get deposited in organs like the heart, or the kidneys, or the nerves, and just start messing everything up. Now, this is a really important point. These amyloid deposits, they're not toxic in the way you'd think of a poison. The scientific term is metabolically inert. They just sit there. The damage they cause is purely physical. It's all about them just being in the way. The best analogy I've heard is to imagine pouring concrete into the delicate gears of a clock. The concrete isn't poisonous to the clock, but it gums up the works and stops it from doing its job. That's exactly what's happening in the organs. Okay, so like any good detective story, there's a plot twist. It turns out there isn't just one kind of rogue protein. Nope, there's a whole lineup of different suspects. And figuring out exactly which protein is misfolding, that is absolutely 100% critical. Why? Because it changes everything, how you investigate, and most importantly, how you treat it. So let's look at the main suspects, the big three for systemic disease. First up, you've got AL amyloidosis. This one's caused by a piece of an antibody called a light chain, and it's churned out by abnormal plasma cells. Next in the lineup is ATTR. Here the culprit is a protein called transthyretin, or TTR for short. And this one can go rogue for two reasons, either a genetic mutation you inherit, or sometimes just from the wear and tear of aging. And finally, there's AA amyloidosis. This type is actually a reaction to some other long-term problem, like a chronic infection or inflammation. See? Totally different proteins, totally different reasons.
It's also super important to get this distinction: systemic versus localized. The three we just talked about, those are systemic. That means the rogue proteins are floating around in the bloodstream and can land and build up in pretty much any organ in the body. But then you have localized forms. A famous example is Alzheimer's disease, where the amyloid beta protein builds up, but it's pretty much confined to just one place, the brain. And look, this isn't just some super rare, abstract medical curiosity. Check out this number. A specific genetic mutation for that HETR type, the V122I mutation, is carried by roughly 4% of American and Caribbean Black people. 4%. That's a huge number. It makes this a really significant and, frankly, often overlooked cause of heart disease in that community.
All right, we've got our clues, we've got our lineup of suspects. So how do doctors actually crack the case for good? Let's get into the detective work.
the diagnosis, and see how they can be sure it's amyloidosis, and most importantly, figure out exactly which type it is.
The definitive proof, the thing that says, yes, this is it, comes from a biopsy. A doctor will take a tiny tissue sample, often just a bit of fat from the belly. They put a special stain on it called Congo red, and then they look at it under a polarized light microscope. What they're looking for is this one specific telltale sign, a beautiful but ominous apple green glow. That glow, that's the smoking gun. Once they see that, they know it's amyloid. The next step is a cool technique called mass spectrometry, which is basically like running a molecular fingerprint test to identify the exact protein.
And I really cannot stress this enough. Identifying or typing that protein is absolutely essential. It is not optional. Why? Because the treatments are night and day different. Imagine giving a patient powerful chemotherapy, which is for AL amyloidosis, when they actually have the ATTR type. It wouldn't work at all, and it would just cause unnecessary harm. You absolutely have to know your target.
You know, for a really long time, getting diagnosed with amyloidosis was, well, it was grim news. But that story is changing, and it's changing fast. So let's shift gears and talk about the modern response, because we are truly in a new era of treatment. These new approaches are turning what was once a devastating diagnosis into a condition that, for many people, can be managed.
So, the foundation of any treatment plan is what we call supportive care. This isn't about attacking the disease itself, it's about managing the damage that's already been done. So that means things like diuretics to help the kidneys handle fluid, being really careful with medications for heart failure symptoms, and using specific drugs for nerve pain or stomach issues. It's all about improving quality of life and keeping the body as strong as possible while the more targeted therapies do their job.
Okay, now for the really cool stuff, the targeted, game-changing therapies. Let's focus on ATTR amyloidosis, because there are two brilliant strategies here. On one side, you have protein stabilizers. Think of them like a scaffold that wraps around the TTR protein and physically holds it in the right shape so it can't misfold. Then, on the other side, you have gene silencers. These are incredible. They go right to the source, the liver, where the protein is made, and they basically just turn down the faucet, massively reducing how much of this rogue protein even gets produced in the first place.
Now for amyloidosis, the plan of attack is totally different because the source is different. Remember those rogue plasma cells? They're the target. The whole goal is to get rid of those faulty factories that are churning out all the misfolded light chain proteins. So the therapies here are often much more like cancer treatments. Think chemotherapy or targeted antibody drugs, very similar to what's used for multiple myeloma. The logic is simple. Stop the source and you stop the disease.
So when you step back and look at it, by figuring out how to crack the case of amyloidosis, science is learning how to deal with this fundamental problem of proteins misfolding. And that leads to a really big, really hopeful final question. As we get better and better at this, at stabilizing proteins, at silencing genes, what other diseases could be next? I mean, think about other devastating conditions linked to misfolded proteins, like Parkinson's or Huntington's, maybe even Alzheimer's. Could the breakthroughs here be a roadmap for them too? It is an absolutely exciting time to be watching this space in medicine.